All right, so let us look at our quiz here. So Darlene, drop o blood. So again, like I said, this is definitely going to be questions that are going to be on your main exam. Might be like Benny the blood drop. Might not be Darlene again, but it'll be a blood drop. And it's always going to be something of this theme. Like the blood drop is someplace and then has a journey to take. And you have to come up with both the vessels as well as if it's going through the heart, different chambers or going past valves, which valves are going by. So for little Darlene the blood drop, where is, so we have her starting in the lungs. So this is the starting point. And so there's going to be a few different steps here. What's going to be the first thing she's going to have to do? Because she ultimately wants to go to the arm. So how is she going to get to the arm if she's in the lungs right now?
She's going to go to pulmonary veins.
Exactly. So we're going to need to go through the pulmonary veins. Those are going to take us back to the heart. Once you're at the heart, what chamber is she going to arrive in?
The left atrium.
So the left atrium, that's where the oxygenated blood is returning. And so now she's back in the heart, but she wants to get out to the arm. So what's she going to do?
Go through the bicuspid valve into the left ventricle.
So it's going to be the bicuspid valve. I'm just going to say valve because I'm trying to write the mouse here, which is awkward. I need to figure out, do this valve, 4, it's going to be into the left ventricle. And then what happens next? What's the next step for her?
She gets pumped out of the aorta.
But before the aorta, what's she going to have to go past?
Aortic tricuspid semilunar valve.
Yeah, so and you just call it semilunar. Even though it does have those 3 flaps, it's not tricuspid. So it's just called the semilunar valve, the aortic semilunar valve. I'm doing this so you can kind of see it, but it's really bad here. So this would be 4, this would be like 6, 7 would be the aorta. And now it's still more. Where's she going next? She's up the aorta, but she wants to get to the right arm. What is going to come next?
To subclavian.
Say it again.
Brachiocephalic.
It'd be the brachiocephalic trunk coming up off of the aorta. I'm trying to minimize. And oh my God, I can actually do this with typing. Duh. Okay, so brachiocephalic trunk. And what's she going to go to next?
Subclavian artery.
Right.
Say that one more time.
Subclavian artery.
Exactly. It's going to be the right subclavian artery. And then where's she going to go next?
Axillary artery.
Yep, that's going to go to the axillary artery. And continuing on her journey.
The brachial artery.
Yeah, so then 11 is going to be, she's in the brachial artery. And at this point, that's all we got since she just has to get to your arm. As I have these kind of questions, sometimes she might be in your big toe and she has to go back to the lungs and get the oxygen and then come back to, So it could very well be there's many more steps involved. Since this is a warm-up, this is just a short little journey. But it could be if she was in your toe and just delivered blood or delivered oxygen, and she needs to go back to the lungs, pick up more oxygen, and then go off to your left thumb. She might have to go back to the heart, off to the lungs, back to the heart, and then off to your thumb. So, these are kind of questions that will be on the exam. So think about blood flow through the heart, as well as how different parts of the body are interconnected by the major arteries and veins. So that is that. Oops, I have to clear that out of the way there because that looks bad. Clear. Okay. Next, here we have a heart. I on purpose did a heart that does not have any color, because the whole point here is I want you to kind of recognize things based on kind of where they're coming from. So we have blood vessel A. What is blood vessel A? We see it's this one of these.
Carotid artery.
So that's going to be, and if you want to be more specific, how could you be?
Left common carotid artery.
It's the left common carotid artery.
Wait, so the right one, the right part of it, is that the right common carotid artery?
Say that one more time.
So the vein right next to it on the right is going to be the right common carotid artery.
Right carotid's up here.
Oh.
Right common carotid and the right subclavian all come off of the trunk.
What's right next to it? What's that? Right next to it. Yeah.
That is going down to your arm.
Brachial artery.
Well, but first, what's the main one that comes off goes under the clavicle here? It's the same name as this one here.
Brachial.
Left subclavian.
Yeah. So this is the left subclavian. Over here would be the right subclavian.
Oh, okay. Thank you.
All right. So those are the subclavian arteries. And then B. What is B?
Pulmonary artery.
Yeah, so this would be, ultimately the pulmonary trunk is the main one coming up and branching into the pulmonary arteries. So I think I gave you credit for either one on that. Officially, B would probably be the pulmonary trunk because it's still where it's one pipe coming up. So those are some of the vessels coming off of the heart. So that was question 2 of the warm-up. Then we can continue. What is the name of the structure of specialized cardiac muscle that you use to coordinate the entire heartbeat?
Vagus.
SA node.
It's the sinoatrial
Atrial.
node.
Yeah, I know atrial node is going to be that.
I think you skipped over number 3.
Oh, I skipped number 3. Oh yeah, so the nerve that slows down your heart. Yeah, cranial nerve number
10.
10. So vagus nerve, the wanderer coming from your head, slowing down your heart. And then finally, we have our little guy with some labeled vessels. What do we call this vessel that's going down his upper arm?
Brachial artery.
That's just going to be the brachial artery.
Can you show where the axillary artery stops on him?
It would be just kind of under, axilla. So,
Okay, so it's like right in there.
Yeah.
Okay.
It basically, by the time you're in the arm, it's brachial. When you're right up, let me get get my little highlighter again. Let me get a color that's not the same color as everything else. You know, right when you come off, it's subclavian, then it's axillary, then it's brachial.
Okay, thank you.
We got to clear. So brachial artery, vessel B, that big pipe coming up towards the head.
Left common carotid artery.
That's going to be the common carotid artery. And see the thing branching off to the bottom of the aorta there?
Right iliac artery.
It's a common iliac artery. So the common iliac artery, which is then going to branch into the internal and external iliac. And then finally, what is D? Just that big hose coming down the middle of the body.
Abdominal aorta.
Yeah, you could just call it the abdominal aorta, or you could call it the descending aorta. Those would be kind of the same thing. So that is the warm-up. Are there any questions about the warm-up for today?
Do you still need to go back and correct these?
This one, yeah, because Darlene is a short answer. So
Okay.
computer does not have any way to know if you've got it right or not.
So I actually like wrote out 10 for the vagus nerve. Is that correct still, or?
Yeah, fine.
Okay, because it marked me wrong. I was just wondering if we should be doing like X or like actual numbers.
Right, so usually it's written as an X. You know, so, and I have to kind of guess what are the possible answers you're going to type in when I do it for the auto grading. But, you know, as I've done a few of these now, I'm finding that there's no way I can ever come up with all of, even if I type in 20 different options that I think you might write and all the probable misspellings that you're going to do, I always miss stuff. So I just always go back manually and regrade everything. This auto grading thing, you know, it's for true and false or multiple choice, then there is no wiggle room. It just knows it's right or wrong. But I hate those kind of questions, as, you know, I think they're useful for some things, but it's not what I don't think, you know, the real world isn't like forced choice all the time. Most of the world is like, wait, what do you call that? And you have to kind of access it on your own without just, well, I'm going to pick one of these four things in front of me. So I'm trying to make sure that the testing is not kind of going to this lowest common denominator of, well, it's just going to be easier to make it all multiple choice, but it means that automation is not as convenient. So, let us get back to cardiovascular then.
Are we going over the other quiz that you said that we just took, or?
We are, but let's talk about some material, new material, then we'll go over the quiz in a little bit. Kind of try to break things up, keep it fresh. Wait a second. I need to stop this. So we are going to continue. I shouldn't even say continue. We're going to finish up. So we have a couple of more things. We have to do a little bit more about blood vessels. And then we also need to get into doing the blood. So let's start with the vessels. Then we'll get to the blood. And normally this day would be kind of more of a review day. So I think it probably makes sense. I think we're gonna be finished with the cardiovascular and the blood. I have been looking at some of the cadaver dissection videos, so I'm gonna show you some of those to give you a sense, particularly of vessels. Arteries versus veins are really different in real life. So showing you some of these videos, I think will try to give you a sense of that. We'll do histology of blood, but then we're just gonna stop there. We're not gonna go on to lymphatic and stuff, even though we should be able to cover this relatively it's not gonna take tons of time. So we'll see how it goes. So let's go back to cardiovascular system. So last we were talking about arteries versus veins. So what are some of the differences that we talked about, arteries versus veins? They're both similar in that they're tubes that are carrying blood. They're both lined with endothelium. They both have this adventitia or tunica externa outside. They both have this tunica media, which has the smooth muscle and elastic, but there's some big differences. How are they different?
Veins are more floppy.
They're way floppier and thinner.
Veins have bigger diameter.
Larger diameter.
Thinner walls.
These have little one-way valves inside. These tend to be thicker. These have no valves. And say it again.
Has an elastic membrane for the artery.
So, I think they both, to some degree, they can have elastic tissue in there, but definitely it's much more prominent in the arteries. And particularly, we talked about there were elastic versus muscular arteries. The muscular arteries have more smooth muscle in the walls just to adjust their diameter, like the brachial artery and most of the arteries that we've been naming are muscular arteries. The elastic arteries are like the aorta and the pulmonary trunk, the major pipes coming off of the heart that can absorb under the high pressure when the ventricles are in systole, when they're contracting. And as they expand, they kind of store some energy. And then when the heart is in diastole, when it's relaxing and refilling, that stored energy in the stretched walls compresses in and keeps the blood pressure from dropping to zero. So the elastic was like the aorta, pulmonary trunk. We talked about how do we make sure that the blood keeps flowing in veins even though the pressure is really low?
Massage veins.
So what massages them?
Skeletal muscle.
So the surrounding skeletal muscle that's kind of moving ends up kind of massaging and pushing the blood along. Um, skeletal, not smooth. Some people, it's normal to think like, oh, there's muscle in the wall, so maybe that's playing a role in propelling the blood, but it really doesn't seem to do. It's the outside, the surrounding skeletal muscle that is massaging and keeping the blood flowing. All right, so we are now going to go, and and I should mention, as you get smaller and smaller, arteries finally, so I should use a small font as well. The small ones are arterioles. When is it an arteriole? It really depends on the book. Somewhere around 0.3 of a millimeter or so, it's an arteriole.
Would the brachial artery be arterial because it's small?
Wait, say that one more time.
Would the brachial artery be arterial because it's small, smaller in diameter?
Oh no, the brachial artery is not small at all. The brachial artery is like a quarter of an inch in diameter.
What would be an example of that?
The arterioles don't have names anymore. They're here, I can let's say here's the brachial artery, and then it has little things. It's going to have little other arteries coming off, which you don't have to know, the circumflex and this coming off of those are little coming up. Once you start getting into the tiny little things that are not quite, they're still kind of delivering blood, but they're just itsy bitsy dinky little things. Oh, come on, Mr. Mouse. I'm going to for you. Give me just a few moments here. Okay, I found it. View image. Resize this a bit. So there's this guy whose name's Gunter Hagens. He does these amazing human dissections, and then it's this thing called plastination. He preserves them, and he makes all these amazing dissections. I've seen a few of his exhibits. They're kind of mind-blowing. And one of the preparations that he does this is like somebody's face where they have injected the arteries with red plastic. So here, this is like if you just injected your blood vessels with plastic and then dissolved away everything else. This just kind of gives you a sense that you've got blood vessels getting everywhere. Like I mentioned, you've got literally 100,000 kilometers of blood vessels to supply every little itsy bitsy part of you. And as those little vascular trees get into smaller and smaller and smaller branches, it's those little small ones. They don't have names anymore. But those are the arterioles. So, anything that we're talking about that has a name is not an arteriole. Those are all arteries. It's the little itsy bitsy branches that continue into smaller and smaller parts of the tree that are the arterioles.
Okay, thank you.
All right, so now it's going to go back to poop. Oh, stop that. So little arteries are arterioles, little veins are venules. And then the business part of the whole circulatory system are the capillaries. This is where materials can enter and leave the blood. Right, so this is where oxygen and nutrients can enter into the tissues. Wastes from the tissues can come into the blood to get carted away, or hormones if you're absorbing hormones, or nutrients or whatever. So capillaries are going to be essential. There are three main, and I should also say one more thing. They're mainly pretty much pure endothelium. Basically meaning they're made out of simple squamous epithelium, right? So they're super thin. Again, they are the thickness of a squamous cell, which is a little tiny frisbee cell. The diameter is really dinky. You know, the diameter is around like 10 microns. So a micron is a millionth of a meter. So 10 to the 6th meters is a micron. A blood cell, so just for a red blood cell, is about 8 microns across. So red blood cells are basically going through here single file. You know, that's going to maximize the ability to transfer oxygen, you know, using diffusion by minimizing the distance. The cell is right near the wall. The wall is really thin, and the cells that you're trying to supply nutrients and all that are right on the other side of that thin wall. So it's kind of minimizing the distance that oxygen has to diffuse and all that. So that's kind of the basics of a capillary. Really dinky little tube made out of simple squamous epithelium. And there are 3 kinds of capillaries you need to know. One of them is the most common, so we'll start with that. So these are kind of your classic capillaries, the ones that are just sending oxygen and nutrients to the tissues and then coming back to the heart. The basic way you would draw that, oh, I need my, let's see, let's make it red. So we have some arteriole, which is kind of entering in. And then we have the actual little capillaries. So I'm going to call my capillary bed. Usually they're drawn kind of blue because as the blood is going through the capillary bed, it's giving up oxygen and going from red to blue. You know, on the other side, we are now in a venule heading back to the heart. The entrance to the capillary bed is a little, we call a precapillary sphincter. So this is used for a couple of reasons. The precapillary sphincter, it can adjust whether or not the capillary bed is open or not. It squeezes. The sphincter is just a little smooth muscle that squeezes. It might be you don't need to send that much blood into this capillary bed. Like, for instance, your muscles. Right now, when you're just sitting here in front of your computer, your muscles only have about 10% of their capillary beds open. Your muscles don't need a lot of oxygen and nutrients to just sit on your butt in front of your computer. However, if all of a sudden you get up and start running around, you can start sending more and more blood into the muscle tissue, open up more and more of these precapillary sphincters, and you can have 10 times as much blood perfusing into the tissue by having a bunch more of these capillary beds opened up. So precapillary sphincters can kind of regulate, how much blood do we actually need to be sending into this tissue at the moment based on its metabolic needs. Another thing is, even if we are opening it up and letting the blood in, the precapillary sphincters tend to kind of pulse open, close, open, close, open, close, open. They think it's partly to kind of keep kind of little jolts to make sure the blood doesn't get caught, right? Because the blood is kind of coming in one cell at a time. The blood cells actually can like start sticking to each other and stuff. So by having this kind of open and close, it's kind of giving it a little shove and keeps the blood moving along to the other side. If the precapillary sphincter is closed, there's still blood arriving here. So we have a kind of a shortcut called a meta-arterial. Maybe let me do it with a bigger. So I'm kind of drawing it in a different color because it's not about delivering oxygen or anything. This is about kind of bypassing the capillary bed so there's some minimal flow from one side to the other. This is called a meta-arterial. So even if the capillary bed is closed, there's still going to be some flow from one side to the other. So that is continuous capillaries. Again, the most common of all the kinds of capillaries. We will come back and see these a bit when we get to the lymphatic system next week. Partly because, blood vessels they have to be kind of porous. The whole point is capillaries allow things in and out, which means they're kind of leaky. We're going to see there's some that are super leaky, but even regular ones are kind of leaky, which means that fluid is going to actually leak out into the surrounding tissue. And that's going to be like one of the jobs of the lymphatic system is to kind of collect lymph, like leaked fluid that's coming out of the capillary beds into the tissues. So we'll come back and see this capillary bed again when we get to the lymphatic system next week. But for right now, we will bid it adieu. Ta-ta, Mr. Capillary Bed.
Professor, I have a question, and maybe this is too complicated to explain right now, but how do the skeletal cells signal that they need more oxygen? How do they signal their metabolic rate?
So one of it has to do with just the amount of oxygen and the amount of the amount of carbon dioxide building up in the tissues. Some of it is what they call like local autoregulation. And then there's also things where your sympathetic nervous system can also adjust blood flow to the muscles and increase blood flow to the muscles, divert it away from the digestive system.
Cool, thank you.
Both central controls and just local auto-regulation controls. All right, so we're gonna do two specialized kind of capillaries. Or I should say capillary types. One of them you've already met. So let's start with that one, the fenestrated capillary. Dagnabbit. So fenestre actually means having windows. A fenestrated capillary, you know, I talked about how a normal capillary is already kind of leaky because it has to let things go in and out. Fenestrated capillaries have little pores, these little windows that make them super leaky. We find these in two different kinds of cases. One is if we have to really absorb a lot of stuff. Like, you know, let's say this is in one of your endocrine glands where the whole job is to be dumping hormones into your bloodstream to distribute to your system, or in your digestive tract where the whole point is to be absorbing all these nutrients that are getting absorbed. So these can be useful for absorption. Or they can also be used for filtration. And let me mention what that means. So in filtration, you actually have, you know, the blood is under pressure in here, and, you know, the fluid leaks out. This is how we made CSF. These were the little dudes that were in the choroid plexuses that we saw in the ventricles in the brain. So the little pores are not big enough to let cells. The cells, the large proteins, they stay trapped in the blood vessel, in the capillary. But you can get fluid and small dissolved things like, you know, salts and stuff like that are leaving and go out. So it's called filtration, you know, because think about like a coffee filter. If you're making a pour over coffee, you've got your coffee, ground coffee inside the filter, and then you're pouring the water over that. And then the water picks up the caffeine and the yummy, you know, kind of flavory things and brown color, which then goes out into your cup. But the big stuff, like the coffee grounds, they stay behind the filter. They would, you know, the blood cells, the large proteins, they stay in the vessel, even though the water and very small dissolved things are managing to leak out. So fenestrated capillaries. We're going to see these again as a key player in the kidneys at the beginning of the nephron, where you're creating this filtrate to help process the blood. So we'll meet these again. The last one I'll mention are called sinusoidal capillaries. So these are going to be found specifically in, these are going to be in the liver, spleen, in bone marrow. These are where the blood kind of goes through really slow, where the blood can get processed. So not only are they super leaky, but there can actually be breaks in the wall. So like white blood cells can reach in and kind of process and clean up stuff. They're very twisty and turny, so the blood slows down, so it gives the surrounding tissue time to process what's going on in there. Like in the liver and in the spleen, this is where if you have worn out red blood cells, these little, these macrophages, these white blood cells will come in and just eat stuff that is trying to clean up and refresh your blood here. So sinusoidal capillaries are only going to be in these very special places where kind of blood is being processed. So they're important, but they're very specific to certain places. So then we're kind of last comment on vessels.
Sorry, I had a question. So for the fenestrated capillaries, where would you find those?
Making up the choroid plexus, for instance.
Okay, got it. Yeah. Okay.
So there is the circle of Willis, which again, it's a classic thing to cover, and it's kind of cool. So we're going to talk about the circle of Willis, which is a specialized arterial anastomosis that is found supplying your brain. So let us do the circle of Willis. The circle of Willis. Boom, boom, boom. All right, so the circle of Willis. First, we need to bring in the brain. Oh, let's make it red. There's the pons and the medulla and the brainstem. So this is brain from the bottom. This is the back. This is the top. But your nose and your eyes would be up there. But so this is like the spinal cord here. So the circle of Willis is going to have the main arteries that we've already talked about supplying the brain, and then things that interconnect them. So name one of the important arteries that supplies the brain?
Carotid.
Totally, the internal carotid arteries. So those are going to be coming in here. The internal carotid artery is going to give rise to both the anterior cerebral artery and the middle cerebral artery. So this internal carotid is coming up from, that carotid canal, right? It's coming up from the floor of the cranial vault. And then as that comes up into beneath the brain, there's a branch that goes forward, kind of right in that crack between the hemispheres called the anterior cerebral artery. Haley's saying that there's a picture in the book. I also have, I'll share a a picture from the PowerPoint as well in a few moments as well. But if you have the book right beside you, right as we're doing this, that probably make more sense as well. What are the other arteries that are coming in?
Vertebral artery.
Say it again.
Vertebral artery.
Exactly, the vertebral arteries. Remember the vertebral arteries coming up from the ones going through the transverse foramina of the cervical vertebrae and then entering up into the foramen magnum of the skull. They merge into a single artery called the basilar artery. And then that basilar artery becomes the posterior cerebral arteries. So that is the initial this is the setup. There's obviously not a circle yet. So we have, let me get fatter thing. We basically have our carotid arteries coming up, going up to the front, and then coming down to the middle. We have our vertebral arteries coming in, going to the posterior. And the circle of Willis is a couple of vessels or a couple of sets of vessels that actually create a circle, a backup. So if anything gets blocked, there's a second way for the blood to get where it needs to go. So one is going to cut across these two right here. This is going to be called my anterior communicating artery. Actually, let me make it a little smaller. So the anterior communicating artery is that little vessel that connects the in between the anterior cerebral arteries. And then we have connections between the posterior and middle cerebral arteries. Those are going to be called the posterior communicating arteries. And what that means ultimately is you got a total circle. You know, I can get all the, even if this was blocked, if this was blocked here, I could still go, woo. I can get wherever I want from either side. So the circle of Willis, just basically, this is an example of an arterial anastomosis. Kind of a backup. And let me show you, I do have actually thought ahead and got this up ahead of time. So this should happen quickly. All right, so it's not quite as endearing as my drawing, but it's probably a little clearer. So again, vertebral arteries coming up, ultimately, initially coming up the little canals on the sides of the cervical vertebrae, then up into the foramen magnum. Now they're up here against the pons. We've got the basilar artery where they kind of merge together. If we still had our if we still had our brains, you could actually see this pretty well. This is all pretty easy to see on the actual brains. This is kind of posterior cerebral, which this isn't a great picture here. This is common, the internal carotid, which goes, yeah, this is now I'm looking at this picture and it's a pretty crappy picture, but you kind of get the idea of it. This is better. Basically, vertebral arteries go into the basilar artery goes to the posterior. Internal carotid becomes the anterior cerebral and the middle cerebral. And then anterior communicating and posterior communicating finish up the circle. So, circle of Willis. You should know it. You should know the circle of Willis. It's kind of a classic anatomy thing. So while we're here, let me see if there's any. I'm going to show you a little more about this on the cadaver dissections, but just kind of giving you a sense of, if you look at this picture of the femoral artery, it looks kind of like a rubber tube. And you look at the femoral vein, it looks kind of this floppy thing that's collapsing in on itself. You can kind of get a sense from looking at this picture. On the cadaver dissections that I'll show you after our break, you'll see even more clearly that, veins and arteries are really structurally different when you actually see them. Like if you take this, femoral artery and you pinch it between your fingers, it'll snap back open. Like it's like a rubber tube. Whereas the great saphenous vein, you might not even know it's a tube until you pick it up and kind of roll it in your fingers and realize, oh, it's actually a tube and not just like some, flat piece of tissue or something. Here's the femoral nerve coming down. This is kind of a classic thing, this little femoral triangle. You have like the nerve, the artery, the vein. There's like, so anyway, let's continue. Not need to know the veins drain this. I don't even know if this has a name, but you don't need, only thing you need to know about the drainage of the brain is ultimately everything drains into what? All the CSF and blood from the brain drain into what major vessel?
The jugular.
Jugular.
Internal jugular vein. We talked about all this. And again, on the little dissection that I'll show you in a little bit, you can kind of tell the difference. Again, when you're looking at your books, you also kind of want to keep in mind some of these vessels are very superficial. When you're looking at the veins, like the cephalic vein, it's just under your skin. When you peel the skin back and you're just trying to skin the cadaver, you have to be careful not to shred the cephalic vein because it's right there at the surface of everything. Whereas the brachial vein is deep and you don't actually see that unless you like kind of dig underneath the muscles and things like that. So cephalic vein, basilic vein are the superficial ones. Again, they connect with the median cubital vein, which is the one people usually take blood from. So you should know the veins of the arms. I mentioned the great saphenous vein as being kind of this big shortcut all the way up to the iliac vein here. If you want to do that. And then this is kind of showing again this idea of the hepatic portal system. Let me get a different color. Like all of these mesenteric veins that are draining the digestive viscera, All of these, they all ultimately feed into this thing, the hepatic portal vein. And again, the hepatic portal vein does not go back to the heart. It goes to the capillary bed number 2 in the liver where the nutrients get processed. And then finally, the hepatic vein is going to drain back into the vena cava. So first capillary bed is in the digestive organs, where you're like picking up nutrients and stuff. The second capillary bed is in the liver, where you're processing stuff. And then you finally send stuff into the vena cava, back to the heart, into general circulation. So make sure you kind of keep track of the hepatic portal system and what it is, and how it's this kind of thing stuck in between the arterial and the venous circulation. This, I actually do want to talk about this depending on the time. There's a few major differences between fetal circulation and adult circulation. One thing, obviously, a baby or fetus is getting nutrients from the mom through the placenta, which we don't got to do anymore. And also, they're not breathing. So they don't need to have this whole pulmonary circuit going online the way we do. And the reason why we're going to want to talk about this is there's a lot of vestigial stuff left over. When we actually look in the cadavers or look at the pictures of the cadavers, you're going to see the remnants. You'll see structures that don't really make sense unless you think about what was in place while you were still in utero. So things that are blood vessels supplying, the fetus from the placenta become structures that are just these little fibrous connective ligaments that are left over, but they make way more sense once you kind of understand developmentally where they came from. So I want to take a detour, I want to go and make sure we kind of get through blood in a more relaxed way, but we are going to come back to this. We'll return to fetal circulation. So let me open up my blood one. So blood, plasma and formed elements. So we're going to look at both of those. So, and I'll come back to the, I'll come back to the PowerPoint in a little bit, but for right now, I think it's better to. So what kind of tissue is blood? Oh, like if you're going to be pedantic about it, what is the type of tissue that blood comes under?
Connective tissue.
Yeah, blood is actually considered a fluid connective tissue where you have, widely spaced cells and extracellular matrix, which in the case of blood is the plasma, the fluid. So we are going to both look at the plasma, which is the liquid, and then the cells. And the cell, it's not just cells. There's also platelets, which aren't quite cells. So there's a more generic term called formed elements, but it's mainly cells. And we are going to, briefly look at both of these. So plasma, what is the majority? Plasma is mainly what?
Proteins.
Water.
Water. It's mainly water. And then it's got, you know, obviously it's got dissolved nutrients and wastes and stuff. It's got the, you know, glucose or whatever that you're trying to deliver to your body. And that, but it doesn't take up a lot of volume. If you think about it, if you have a glass of coffee and you put in a spoonful of sugar, it doesn't really make the, you know, volume change much. So maybe 1% is going to be dissolved, you know, dissolved nutrients and wastes and hormones and stuff. And then that's going to leave us with, you know, 7%. And then I heard somebody say these are going to be what we call plasma proteins. So plasma proteins do a variety of jobs. What's like there's alpha and beta globulins. There's albumin. So let's talk a little bit about what they're doing. So one thing that they can do, if you want to transport something in the blood, it has to dissolve in water, right? You know, plasma is water, but there are things that you want to transport in the blood that don't dissolve in water very well, like steroid hormones don't dissolve in water. There's a bunch of vitamins that are lipid-based, fat-based that don't dissolve. But if there are things that you want to move through the bloodstream that don't dissolve in water well, you can have special plasma proteins that help bind and help carry them through the plasma. So they can be what we call like, you know, they can be carriers to bind and move things that don't dissolve well in water. Again, like a classic example is a steroid hormone. Steroid hormones are constructed from cholesterol, a lipid, and they're not going to just dissolve if you just put them in water. So this is to, you know, help carry, you know, what we call like, you know, kind of, you know, things that are hydrophobic or things that, you know, lipophilic, things that don't dissolve. Also, it's going to deal with osmotic pressure. If you remember osmosis from your Bio 110, you know, if I have something that's really, let's say, in fact, let me do it this way. Let's say I have my blood vessel here. Let's say it was just, you know, just distilled water. And then we had blood cells in there and we had blood cell, you know, tissue cells outside. What happens to cells if you put them in just pure water? What does osmosis do? People should remember from your Bio 110.
Use water across the membrane.
So the water, so if I have distilled water on one side and something really salty on the other side, which way does the water go?
Towards the salty side.
The water will go towards the more salty side. So, you know, you'd have water just leaving the blood vessels and going into the tissues. So like this albumin in particular, this is like, I think something like 60% of the plasma proteins, helps with the osmotic balance by making sure there's stuff dissolved in the plasma so the water isn't going to want to enter or leave the bloodstream, because the plasma itself is kind of osmotically balanced with the rest of your body. all of these a lot of the other proteins, we're going to see some in a little bit that help with blood clotting. Other ones are actually important for doing body defenses. Other ones are going to be important for controlling blood pressure. There's all sorts of blood plasma proteins that are doing all sorts of different jobs in your body. And that's not we don't have time in this class to talk about them all. I'll leave it there. Lots more doing lots of important jobs. And these are all made in the liver. We're going to talk about the liver when we get to the digestive system, but the reality is the liver does so many different things, including making the plasma proteins. That's one of its jobs. There's one specialized plasma protein not made in the liver that I should mention it for completeness here. I'm going to say also I'm going to make this smaller here. Also gamma globulins. Which are also known as antibodies. Made by lymphocytes. Ooh, let me make sure you can read it. So these are all made in the liver. But if we're going to be complete, there are some plasma proteins not made by the liver, these antibodies, which are made by lymphocytes that are part of your body defenses. I'll talk about these a little bit in a few moments, but we're not going to go. This class, we don't go into depth into the immune system that way. So that's plasma. We have carriers like these alpha and beta globulins help carry things that don't dissolve in water. There's lots of other ones as well. Formed elements, this is mainly their cells. Mainly our blood cells. And then also these little things called platelets, which we'll talk about. So we're going to talk about the formed elements now. So the formed elements of your blood. The most common is just the RBC, the red blood cell. Also known as the erythrocyte. Erythro just meaning red. You'll see that erythro prefix used like erythematosus, like if you have reddening of your skin and stuff. Erythro just means red. Erythrocyte means red cell. Most common, like 99% of your blood cells are RBCs. They are the ones that are carrying oxygen around. So these, let's look at an RBC. From the side, it looks kind of like that. From the top, it looks more like that. When it is developing, it makes lots and lots and lots and lots of this protein called hemoglobin. It fills up with hemoglobin. When it's done filling with hemoglobin, it actually ejects the nucleus. There's no nucleus in this thing. it actually has very little kind of metabolic needs. So it doesn't even have the main, what organelle would it need if you wanted to do like aerobic respiration? If you wanted to make ATP using oxygen, what's the main organelle you would use?
Mitochondria.
Yes, exactly. And these guys, they have no mitochondria. Which kind of makes sense. Their job is to transport oxygen, so you don't want them kind of using up the oxygen that they're supposed to be delivering someplace. And the other thing is they have about 250 million molecules of hemoglobin. So a whole lot of hemoglobin. Hemoglobin is a special protein. It's actually got multiple parts. It's got this little heme group. Contains, so this is my hemoglobin. One, it contains iron. So you have to have iron to make hemoglobin. And each hemoglobin can bind 4 oxygen molecules. So what does that mean actually? If we talk about there are 250 million molecules of hemoglobin in one cell, and each cell can bind 4 oxygen molecules. How many oxygen molecules can you get in a single red blood cell?
A billion.
Yeah, if I was think of like Dr. Evil. How many oxygen molecules? 1 billion oxygen molecules. So you've got an amazing amount of ability to carry oxygen around. That's their job. Their job is picking up oxygen in the lungs and delivering it in the tissues. So the red blood cells do that. If you have no nucleus, what does the nucleus usually have? What's the use of having a nucleus?
Stores genetic material.
Replicate.
It's storing all the genetic material. It's like the blueprints. And so if you don't have the blueprints, if things start breaking down, you can't really fix things. You don't have the plans to like, make new parts and pieces and repair and replace things. So these red blood cells wear out pretty quick. These only live like 3 to 4 months. So they are circulating around. They have some minimal amount of metabolic activity. They can use glycolysis to make ATP. They don't need mitochondria. But after 3 or 4 months, they've worn out and they need to get recycled. They end up getting recycled in your spleen and your liver. Maybe I can put that on here. So we talked about how these sinusoidal capillaries are places where blood can get processed. So these are places where you have like white blood cells and stuff reaching in and, scavenging worn out red blood cells and recycling them. The waste product of, it's kind of fun to think about. One of the waste products of recycling the heme group. The heme group is the part that contains iron. This is called the heme group. Part of recycling that, you end up with this stuff called bilirubin, which ultimately is getting dumped into your digestive tract, and it's going to ultimately be the source of what's coloring your poop brown. So it's got that going for it. So blood cells, and as I mentioned before, a red blood cell is about 8 microns in diameter. That's an ugly picture here. So it's kind of like the yardstick of histology, sometimes they call it. Right, because there are red blood cells everywhere. Pretty much any tissue you're looking at, if you are really looking hard, you're going to find red blood cells. And as soon as you find red blood cells, you know about how big they are, and now you have a scale for everything else around you without having to do any calculations or anything. You know, like, oh, if this thing is a little bigger than that, then I know it's on this order. If this is a useful thing to know. It also, if I think at this point, we're probably not going to be trying to find capillaries. I am going to talk about arteries and veins under the microscope when we get to the lab time. But a capillary, because it's about the same size, I said it's about the same size as a red blood cell. They go through single file, and they're made out of simple squamous epithelium. You know, capillary is tiny and made with super thin walls. So when you are actually looking at tissue with all sorts of stuff going on, capillaries are just going to be itsy bitsy things. You know, they're really not obvious. You have to really look around. Sometimes you get lucky and you actually see a blood vessel inside one. And then you're like, I know for sure I'm looking at a capillary because it caught a blood vessel in there. Not blood vessel, a blood cell in there. But capillaries are really, really kind of tricky to find. I think, you know, if we were working with actual slides where you can move around and zoom in and double check things, then it's fun to try to go hunt for them. But they are not so obvious to look at like other things that we've been talking about, little capillaries. You know, blood vessels that are larger are more obvious. Like you'll see, if it's an artery, you'll see nice layers of smooth muscle in the wall. And usually you'll see blood cells inside. You know, if you see something like, I think that's a blood vessel. It's like you look inside, it's like, oh, it's absolutely a blood vessel. I can see the red blood cells in there. So looking for red blood cells is another way also to decide whether you're looking at a blood vessel, because they often end up getting or becoming part of the slide. So it is 2:04. So let us take our break. So wandering through the desert here. Right, so what I'm gonna do now is just finish up with the blood. We still need to cover the formed elements. You know, for the purposes of anatomy, we will talk very briefly about the functions, but mainly we learn about how to recognize what they look like. So let's do it. Again, we're doing formed elements. We talked about, 99% of all of them are RBCs. So we kind of finished. So that's most of the blood cells. When we look at blood under the microscope, it's mainly going to be red blood cells. But then there are the white blood cells. So that's what we're going to talk about now. Again, it's also there's WBCs. And they're also called leukocytes. So white blood cells, leukocytes, WBCs. And these are pretty different from the red blood cells. One is they have nuclei. So when you look at them under the microscope, they will all have a nucleus. Another thing is these are all involved in the immune system. So they're going to be playing a role in protecting your body. They're not carrying nutrients or doing anything like that. And then another big difference is they are not confined to the closed circulatory system. Right, we've already kind of talked about cells that you find in connective tissue in the very beginning of the class. We found lymphocytes and macrophages and stuff like that. Those are white blood cells. So they can move around through your bloodstream, but then they can go and they can ooze out of the blood vessel and get into the tissue spaces. A lot of them just live in the body, moving around, patrolling, like through the, the epidermal macrophages and the layers of your epidermis even. You have other ones that live their life just hanging out in the liver. Other ones that are just going in, hanging out in lymph nodes and then traveling through the lymphatics into other places. So they're all over the place. They're definitely not just stuck in the bloodstream like a red blood cell. There are two main classes. And this I should probably do as a, there's going to be the granulocytes. And there's going to be the agranulocytes. So let's start with the agranulocytes. This is going to include our lymphocytes. Lymphocytes are part of specific immunity. The best way to kind of describe this is some of your body defenses are more general. Like if you want to keep somebody from hurting you, you might put up a fence or put locks on the door. And those are going to just keep anybody out. They're not about any one particular person. Lymphocytes, on the other hand, are looking for a very specific looking thing. So it would be like if you had a restraining order, right? It's like, it's not just about, keeping people away from you. It's about keeping that particular person away from you. The lymphocytes end up getting primed in order to recognize and attack very specific things. Like these are the ones that are, like if we ever find a COVID-19 or not, yeah, COVID-19 vaccine, it's going to be stimulating these guys in your body to recognize the novel coronavirus. So you'll be safe from it because they'll be looking just for the novel coronavirus. So lymphocytes are very specific in how they approach keeping you safe. They look for a very particular thing. So that's lymphocytes. In terms of what they look like, so I should mention when you stain blood, there's this really kind of classic way of staining blood. So stain blood cells with, it's called Wright's stain. And normally I tell you don't use color as a way to recognize what you're looking at. But with blood cells, you do use color to recognize what you're looking at. So a lymphocyte, for instance, a lymphocyte tends to be relatively small, a little bigger than red blood cells, but about the same size as a red blood cell. Then they have a nucleus that fills almost the entire inside of the thing. So there's usually like, a little halo of cytoplasm around it, but the nucleus stains dark blue and takes up almost the whole inside of the cell. The other kind of agranulocyte are the monocytes. So 1, 2 are the monocytes. These things are the ones that turn into macrophages once they leave your bloodstream. So these, the formal thing we would say, they differentiate into macrophages. So once they leave your bloodstream, they turn into macrophages, the big eaters. And they're big. I'm going to tag nabbit. I'm going to get rid of the red blood cell here because I don't need it here anymore. Instead, I'm going to have my macrophage, my monocyte. The monocyte is really big, and the nucleus tends to be kind of bent. Like if you kind of lived in the Hollywood Hills and had your big kidney-shaped swimming pool in your yard or something, it looked like the nucleus of a monocyte. Under the microscope, again, it's not going to matter so much because I'm just going to find pictures that look pretty for you. But in reality, these are big enough that they often get really distorted when you're trying to find them on an actual slide. So in the future, when you're looking at real blood, these can end up getting pretty distorted looking, but I'll just give you slides where they look pretty. So agranulocytes, and these macrophages, big eaters, they eat things. And in fact, they eat things, and then we'll talk to the lymphocytes and tell the lymphocytes, look what I just ate. You better make an clone army to attack this thing that I just found. So macrophages, they eat stuff, plus they can actually talk to the specific immunity and set up body defenses against the things that they caught. Granulocytes, on the other hand, have little granules. There's going to be 3 kinds. The most common are going to be called neutrophils. Sometimes they're called PMNs. or we'll talk about why polymorphonuclear cells. There's going to be eosinophils. And there's going to be basophils. Each of these have little granules. That's how they get their name. Although the granules aren't so obvious. In the neutrophil, the main thing that makes them really obvious is a really weird multi-lobed nucleus. So the nucleus sometimes it doesn't even look like a single thing. It looks kind of like, this would be a classic looking nucleus for a neutrophil. So that's why they end up becoming PMNs. I mean, polymorphonucleus, it means they've got many shaped nucleus. They have granules, but the granules are pretty light. Kind of little blue and red dots, but they're not that dramatic. The main thing you see when you look at a neutrophil is that crazy, weird nucleus. Eosinophils and basophils, and I should say in neutrophils, these are phagocytes. And macrophages are phagocytes as well, but the neutrophils are kind of more like the infantry. They're more like front lines. They are fighting, they eat bacteria, they die. They're a big part of, like, when you've got an infection and there's all that icky white pus, it's like dead and dying neutrophils. Monocytes, the macrophages tend to be kind of more the more kind of elite officers of the immune system. They live much longer and they eat things and then go alert other cells in the immune system. Whereas the neutrophils just go to the front line and eat bacteria until they are filled with toxins and die. So they don't get, it's not as I don't know. They don't have as much glory as like a macrophage, I think. So eosinophils and basophils both have a nucleus that looks kind of like they call it bilobed. So bilobed nucleus. I used to, when I first started teaching, I could just say, yeah, think about like a telephone handset on your telephone. That's what people used to have real telephones. Anyway, the granules are what really give these away. Eosinophils they're filled with these big red granules. Eosinophils seem to be mainly about like multicellular parasites, like worms and things that get into your body. Basophils are filled with blue granules. These are pretty much like the mast cells that we saw in the connective tissue. These basically release histamine, which will promote inflammation. So neutrophils are by far the most common. I can give you the little mnemonic people use. So they say, like, never, how does it go? Never let monkeys eat bananas. And that is giving you the different white blood cells from most common to least common. So N neutrophils, when you look at a slide of the blood, you pretty much always find neutrophils. You pretty much always find lymphocytes. Monocytes you do find as well. Like I said, they often look distorted. You can look quite a long time before you see eosinophils, unless this person is actually actively combating something. Obviously, then there's going to be a lot more eosinophils. But if their body is not actually in some condition where it makes sense to ramp up production of them, they're pretty rare. The basophils are even rarer. I mean, I literally go for years of looking at blood slides and don't actually see a basophil if I'm not really looking for it. I'm going to share the PowerPoint from Marib. So like here's like a blood smear. Red blood cells just look like little red circles. They tend to look lighter in the middle because they have when they eject their nuclei, they kind of suck in. So they look kind of like flat with the middle sucked in. This is showing the different formed elements I've just been talking about. So if I go to my highlighter, neutrophil, it's got this weird looking nucleus. I'll show you more examples of it, but it's always weird looking. There are granules, but they're sometimes hard to see. Here's an eosinophil. You can see the red granules are super predominant. Basophil is filled with the blue granules that are kind of masking the nucleus even there. The monocyte, actually you can see it's getting kind of distorted here. This should not, it should be more roundish, but this is a monocyte with its big kind of like kidney shaped nucleus. And here's a classic lymphocyte. Whole center of it is pretty much a big round nucleus with a little bit of cytoplasm around the outside. You should be able to look at what? Hold on. I have other. Well, maybe there's no more. I'll find more pictures for you of that, so you can try to recognize it on your own. Normally, yes, on a test, I could imagine like having a. I wonder if I can do this. If I put this, yeah. Here, I'm going to go into Google. What's going on here? Yeah, like this is a classic blood smear here. So you should be able to look at this and you can say like, oh, here we've got a neutrophil with all this weird looking nucleus. Here's a lymphocyte with the nucleus that totally is filling the inside up there. Here's another one. Again, beautiful looking neutrophils. And this, that looks like a basophil. That's actually pretty rare. Here, this is a classic again. Lymphocytes, basically a big nucleus with a little bit of cytoplasm on the outside, and then a neutrophil, this thing that has a weird multi-parted nucleus with a little bit of granules. Well, the granules are not that predominant. So hopefully that kind of makes sense there. So think. And let me release this, and I want to. There's one other kind of formed element we should talk about. Then we'll be done with the blood. So remember, we had the RBCs, and we had the white blood cells, and the white blood cells were the neutrophils, lymphocytes, monocytes, eosinophils, basophils. But then there's one last thing that's called a platelet, which isn't really a cell. So let's look at the platelets. These are for what we call hemostasis, which means stopping the blood from leaking out if you have a tear in your blood vessel. So platelets basically are monitoring the structural integrity of the blood vessel. I'll talk about that in just a moment a little more, but first let's talk about them just in terms of what they are. In the bone marrow, in fact, where is blood getting made? I just kind of said it.
Bone marrow.
The bone marrow. So hematopoiesis. What? This is the fancy word for blood formation. And right now I think I missed. No, I think I did have it right. So this is in the red marrow of your bones. You have cells that can make all of these other blood cells we just talked about called hemocytoblasts. These are basically the blood stem cells. And then there are also these big cells called megakaryocytes. So a megakaryocyte, it sounds like a huge nucleus, which is true. The megakaryocyte is this crazy cell, and it makes little vessel-bound bags of granules. So these are the platelets. The platelets are these little things that it's pinching off, filled with little granules. So little vesicle vesicle bag with granules. The granules do all sorts of stuff. I'm not going to get into it right now. There are going to be things that help cause the blood vessels to constrict and attract other platelets and initiate blood clotting and stuff. So they're filled with all sorts of goodies that you would need to start the blood from, to stop the blood from leaking out of the blood vessels. So platelets, but again, they don't look like blood cells. When you're looking at the blood under the microscope, you're going to see, oh, here's all my blood cells. And, you know, maybe here's a neutrophil with its crazy nucleus. The platelets are going to look kind of just like schmutz, like little bits of stuff in the spaces between. The platelets don't look like a cell. They're little bags of granules. And I'll talk a little bit more about platelets. So these basically are triggered by exposure to collagen. So if a blood vessel is intact, if a blood vessel has not been damaged, what is the only kind of tissue that the platelet should see?
Epithelium.
Exactly. It should just see simple squamous epithelium, the endothelium. So as long as everything is cool, they will not see collagen. Collagen is in, you know, it's the fibers of connective tissue. So if there's been a rip in the blood vessel, then it's the platelet that then notices like, oh my God, there's collagen. It would be like, again, if you're inside a room and all you see is paint and wallpaper, you know that the walls are probably intact. But if you're looking at the wall and you're seeing like splintered 2x4s and conduit pipes and stuff, it's like, oh, it looks like there's been some damage to the wall here. So when a platelet gets exposed to collagen, that's the trigger that there's been damage. And then again, I'm in the interest of time and physiology, we go into the details, but it does a bunch of different things. It makes a platelet plug. It kind of sandbags the place where there's a leak. Then it sends out serotonin, which constricts the vessels and makes them kind of squeeze to stop the blood flow to the region. And it initiates all of the blood clotting to create the little blood clot to make kind of a Band-Aid. Then it has growth factors to actually stimulate the smooth muscle and connective tissue and epithelium to all regrow and just repair the vessel back to the way it was. So these platelets are doing all sorts of stuff. And your blood vessels are ripping all the time. You just get, you know, think about it, you get a bruise, you hit yourself, just moving around. These blood vessels are so delicate. So you constantly have to be able to repair the little rips. When people talk about drugs that are that are blood thinners, they actually don't thin your blood. Instead, what they're doing is they are anticoagulants. They're things that inhibit the action that the platelets are initiating. You know, so if somebody you're worried that their blood is too likely to make clots, you can give them things that inhibit this whole system. The bad thing is if you give them too much, like, you know, Coumadin or some of these blood thinning medications, you have to be really careful about the dosage. If you have too high a dosage of blood thinners that inhibit this whole blood clotting thing, then you get bruised just from like your socks. You know, anything just pushing against your skin is making little rips and tears in vessels that normally would just instantly seal up. But if you inhibit that over too much, you get bruised all over the place. So any questions about blood? We just went through a bunch of blood stuff.
I have a question. Does it work the same for artery? If you got ripped, does the platelet stop it?
I'm sorry, I didn't hear it. Say that one more time.
If the artery got cut, does the platelets get stopped or how that?
If you get a cut, the platelets are going to be an important part of stopping the bleeding.
For the artery too? For the artery?
I mean,
For
if,
the artery?
I mean, if you cut a large artery, then it's a different story. A large artery is like a hose under high pressure. And so that's going to be a little tricky. You're going to be spurting blood out of that. But in general, when you have a blood vessel has damage to the wall, the platelets will initiate a bunch of different processes that will heal the damage. So, right. But yeah, if there's like there's a big, if there's a major tear, it could be a little trickier.
Okay, thank you.
All right, so the last thing I'm going to do, I think it's the last thing we're going to do for the cardiovascular system. Then I'll go into just show you some lab stuff, not new material. I'll show you pictures so you can just it won't be more writing. But I do want to talk a little bit about the fetal heart. So what are the big differences between the circulation for a fetus and the circulation for a postpartum human?
They're using a secondary oxygen source.
Yeah, so one thing is, they get oxygen from mom. So they are not going to need to be spending all this energy to spend, half of their blood flow going to the lungs, because the lungs aren't doing anything. The lungs are just waiting till you're born. The lungs need a little bit of oxygen because they're cells that need to live, but you can divert most of the blood that's going to go to the lungs and just send it back to the body. And we'll see that happening. What's another big difference between, it's part of this. So you get oxygen from the lung. So we're going to bypass pulmonary circuit. And also the place where we're getting the oxygen and the nutrients and everything from our mom is from the umbilical arteries and veins that go to the placenta. So there's going to be an umbilical circulation to the placenta. And then one other thing is, because we talked about all of that, the hepatic portal system about processing your nutrients and everything, but you're getting pre-processed nutrients from your mom. You're not eating when you're in utero. So you're also bypassing some of the liver circulation. So these are these three big differences. We'll look at them one at a time. This is supposed to be a fetus. And fetus is in the uterus. And we'll talk about this more at the end of the semester, but you have this thing called the placenta. A woman who's pregnant has a placenta. And the placenta is a place where the fetal blood, so the fetus has these two umbilical arteries that go out. They start at the right near the iliac, the common iliac. So these come off of the iliac arteries. So I should actually have it coming down from here. And then they return in this big umbilical vein. And I'm drawing it blue. It's actually red, right? Because it just picked up oxygen from your mom. But for whatever reason, I just went by my old force of habit and drew a vein as blue. The umbilical vein. And so this is fetal blood. And then maternal blood is coming in here as well. I'm just going to and things to remember here is that the fetal blood and the maternal blood, they don't mix or anything. There is just this place to exchange materials. So the fetal blood can get rid of wastes and carbon dioxide, and the maternal blood can pick it up and take it away. The fetal blood can absorb oxygen and nutrients from the mom. Again, there's no mixing. It's just exchanging materials between the two circulations. The umbilical vein, after you're dead, not yet, after you're dead, my God, after you're born, the other side of dead, after you're born, you can still see what it used to be. It becomes the ligamentum teres, which goes right into the liver. And we'll talk about it in more detail when we do the digestive system. I mean, this, I wish, like, we dissected that big guy really well. So if you you can still have the bit of skin that has his belly button, and you can see the other side of the belly button. And this is on all of you. I don't recommend trying this at home, but if you cut yourselves open and you look on the inside of where your belly button is, you'll actually see this connective tissue cord, which just goes still connected into the liver. And it's the remnants of what used to be the umbilical vein, bringing the blood back from the placenta when you were still in utero. So again, we'll come back to the ligamentum teres, and I'll show you pictures of it and stuff later, but just to kind of get you thinking about it at this point, it's there because it used to play a really important role, even though now it's just a little connective tissue string. So umbilical circulation to placenta. Umbilical arteries and veins going out and out and back, exchanging material with the mom's blood. Then we can look at this, the oxygen coming from mom, so we can bypass pulmonary circuit. So normally you would have the deoxygenated blood, returning into the right ventricle, going to the left ventricle. No, oh my God. Normally you'd have the deoxygenated blood coming back into the right atrium, going down into the right ventricle, and then going out the pulmonary trunk to go to the lungs. But since we don't want to send all this blood out to the lungs, there's going to be two things that short circuit that. One is going to be right across the atria. So there's going to be a little, oh, there it is. Basically a little hole. So the blood that's coming into the right atrium can just come across onto the left atrium and just be back in systemic circulation. So that little dude there is called the foramen ovale, and it just interconnects the right and left atria. After you are born, obviously that has to be sealed up. So you can still see a little divot where it used to be. So it becomes the fossa ovalis. After birth. Wait, foramen ovale. So that's one thing, foramen ovale, which is open. And if blood does make it down into the ventricle and comes out the pulmonary trunk, there's another short circuit right here going from the pulmonary trunk into the aorta. So the blood that is coming out this way can go back into the aorta instead of continuing on to the lungs. That has a name as well. That's called the ductus arteriosus. And that's again a short circuit that goes from the pulmonary trunk to the aorta. After birth, it becomes, again, a little bit of connective tissue that you can see connecting the pulmonary trunk and the aorta, and it becomes called the ligamentum arteriosum. So that is the main stuff you should know. There is also another thing, a bypass in the liver that's hard to see, the like ductus venosus. But the stuff you should know, you should definitely know fetal heart has a foramen ovale, which becomes the fossa ovalis to short circuit the atria. The ductus arteriosus, I think it's a U, becomes the ligamentum arteriosum afterwards. So we still want a little blood going to the lungs because the lungs need some oxygen because there's still tissue that needs to be alive. But this is making it so most of your circulation stays on the systemic side because you don't need to be sending blood to the lungs. And and the other one is the fetal circulation. You are sending blood out to the placenta, having it return, and at the placenta, it's where you get to exchange stuff with the maternal blood. You get to absorb nutrients, give off wastes. So are there any questions about all of that?
I'm just curious, like how long after birth is the like foramen ovale still there? Like when does it become the fossa?
So in general, so there's a couple of answers to that. One is the pressure changes when you were born. Like once you start using your lungs, it changes lots of different conditions inside your body. And some of the changes of pressure that happen is kind of like this flappy kind of thing that will flap over and just block it off immediately. In fact, I had a student where they found that her fossa ovalis never actually totally sealed. It was actually just still kind of floppy and loose, but it was just the pressure was keeping it sealed shut. But in general, just immediately due to pressure changes, it seals over. Some people, though, there's a lot of birth defects where it does not seal, and then you have like leakage that has to be surgically fixed. So the question, yeah, so that's I guess how I could answer it.
Thank you.
Any other questions about all this stuff?
So is that what they would call like a heart murmur?
A heart murmur is typically using the regular valves, right? Not for most people, the fossa ovalis is just sealed off. A heart murmur, there's two kinds that you can get. Heart murmurs can be either because the AV valves don't seal well and the blood is kind of splooshing back up into the atrium when the ventricles contract, and it can happen on either side. And you can also get heart murmurs if the semilunar valves don't seal well. So when the heart is relaxing and you've got all this diastolic pressure here, you can have blood starting to smoosh back into the heart through the semilunar valve. So you can get heart murmurs either from semilunar or atrioventricular valves, but that's typically what they are. All right, so while you were all on break, I was desperately making new parts to this week's module, which I'm going to show you right now. Where did it go? There it is. All right, so if you look at remote week 3, there is now a new module. One says histology vessels. So this is I just basically went into Google Images and found images that I thought looked kind of nice. At the top, these are just the files, and below I just have the pictures, but these things up on here and the pictures down below, they're the same thing. And these are nice to kind of really show you arteries versus veins. And again, this is something you are going to be responsible for. You know, and here I was kind of mentioning that you typically will actually see the blood cells, like here, these little things in here, this is all full of blood cells. You know, here we can see arteries have much thicker walls. They tend to hold their shape better. Veins have much thinner tunica medias and they tend to be floppier. Here again, very obvious. The artery, you can see the smooth muscle, really nice smooth muscle. The elasticity in the thing is making the simple squamous epithelium look all kind of crinkled up here. And here you can see some red blood cells. We continue down again here on this picture. I put this one in here just because it has really obvious red blood cells in it. You know, when you are looking at real slides of real tissue, you know, you're always kind of looking for red blood cells because then you know, like, okay, I know I'm looking at a blood vessel. And here's a classic thing of artery, vein, and nerve. You know, this would be a totally fair question to put on an exam, is a picture like this and be an arrow on one of these structures, and you have to be able to tell, is that an artery or is that a vein or is that a nerve? And how do you know? You know, how do I know that this is an artery?
Maintained.
It's maintaining its shape and it's got that thicker
wall.
Media, this thick, smooth muscle making its wall. This vein is larger, it's floppier, it's the tunica media, you can barely even see it. It's mainly just kind of the connective tissue out here. The lumen is all collapsed in on itself. How do I
see any?
Go on? Jamba?
I don't know. I was just going to ask you if we could see a valve in that vein, a one-way valve.
No, because that would be, you'd have to see it from the longitudinal view, right? You can't really see that on a cross section like this.
Okay.
You know, if you're looking at a vein from the side, the valves are looking like that, but we're looking just at a cross section. So we're not actually seeing this structure in there. Nerves are often in the same region. Like, how do I know that this is nerves and not blood vessels?
There's no lumen.
Yeah, big part of it, there's no lumen. So no lumen, it's a giveaway. And if you look at more detail, it's like, oh, it looks like little itsy bitsy donuts in there. Oh my God, those are like cross sections of axons. And these are actually probably fascicles with perineurium between them or whatever. So that is something when you are doing your studying, one of the classic slides are called nerve, artery, vein. And you should be able to recognize at this point a nerve versus an artery versus a vein. You know, sometimes it's a bad slide or it can be trickier to tell, but the best slides are going to be very clear. This I put in here, I was kind of telling you how it's really tricky to tell a capillary sometime. You know, so this is showing arterioles again, this is looking on a really small scale. You know, a capillary is literally just the size of a blood cell. You know, unless you really know what you're looking for, you wouldn't even know it's a blood vessel because it's got super thin walls and it's super tiny. Arterioles, you can tell, have a little bit of smooth muscle in their wall. You know, this thing over, let me get my annotator going again. You know, if I was wondering what is this, I know it's probably a vein. It's all floppy, thin walls, and it's full of blood cells, right? So I was saying, look, and here again, here's another little blood vessel. You can tell because it's got blood cells in it. Seeing blood cells inside things is often, sometimes they leak. Sometimes during the cut, you just get blood cells leaking all over the place. But in general, they stay pretty well behaved when someone's making a good slide. So you want to look at these and really start feeling that you're being able to recognize an artery, a vein, and go back to the nerves, recognize a nerve as well.
Professor, do you want us to be able to recognize capillaries for the test?
No, like I said, unless you actually are sitting at the microscope yourself and zooming in and out and focusing and up and down, it's really hard. So yeah, you will not be able to, you need to know about them intellectually, but you are not going to be held accountable for them.
Okay, thank you.
Let's see. Again, this is just showing, you know, tunica media, adventitia, kind of. So anyway, I put this up there just so you have some stuff to look at. And then the other thing I put up, is a variety of these cadaver dissections. I was kind of going through them quickly, trying to find, I was just looking at this a little before class as well. So cadaver videos, cardiovascular. And these are these Wisconsin ones again. And I think it's worth spending a little bit of time because you really will get a little bit more of a sense of what a vein and an artery look like in reality, like the floppy vein, the more kind of resilient, rubbery looking artery. So I'm going to start showing this thing. This is looking at the arm. This is the one we looked at when we wanted to see the, it's the same video that you looked at for the brachial plexus, but now we're focusing on the veins. So if we like go in here and this takes a little while, but I think it's worth seeing.
The next dissection is basically skinning the upper limbs. During this skinning process, the only things that we want to make sure that you find are some of the cutaneous veins that will be coursing under the skin and draining the cutaneous surfaces of limbs. There are two vessels in the upper limb that you need to be aware of. One is the cephalic vein and the other is the basilic vein. The cephalic vein drains the anterior and posterior aspects of the lateral hand and basically courses along the lateral border of the forearm and arm to the point where it drains into the subclavian vein up here in the neck. The basilic vein drains the medial anterior and posterior borders of the forearm and hand and runs along the medial border and then ultimately drains into the brachial vein along the medial side of the arm. There's a vein that joins between the cephalic and the basilic vein known as the median cubital vein, which is generally found right in the region of the elbow and is commonly used for taking blood. You should be aware that there are also some cutaneous nerves that travel with these veins, which we're not going to spend a lot of time finding, but they are branches of the brachial plexus that will supply cutaneous innervation to the skin along the approximate same distribution. To take the skin off of the arm, forearm, and posterior or dorsal surface of the hand, you'll make a longitudinal cut on the sort of anterolateral surface of the arm and continue it down onto the forearm, and then undermine the skin, and using your fingers, you can peel the skin off in one big swatch. Watch for the position of the cephalic vein in the underlying surface of the skin, and once you find that cephalic vein, you can just make sure that you don't destroy it as you remove the rest of the skin. After completion of the skinning, you should be able to find the course of the cephalic veins and the basilic veins, along with their associated cutaneous nerves. The cephalic vein drains into the subclavian vein by forming a groove between the pectoralis major muscle and the deltoid, and you can see that it courses along the anterior lateral aspect of the arm and forearm down to the lateral aspect of the hand. There's a small branch between the cephalic vein and the basilic vein that courses right across the median cubital fossa, known as the median cubital vein. This vein is important because it's commonly used for drawing blood. The basilic vein on the medial side of the arm can be seen here as it dives deep into the arm, draining into the brachial. And you can follow the basilic vein down as it continues on the medial aspect of the forearm. Both of these cutaneous veins travel with some cutaneous nerves. The nerve that travels with the cephalic vein is the left.
Right. So we don't care about the nerves, but so there you can see those superficial veins are very superficial. They're just under the skin. The arteries now, so I have here a number 11. I'm going to go ahead to minute 11.
Now that we've looked at the branches of the brachial plexus, what we're going to do on the other side is cut away the things that are made up of the anterior division component of the brachial plexus to uncover the brachial and axillary artery that lies underneath. So we're going to uncover the artery underneath by pulling those branches away. Now we're going to be concerned primarily with the axillary artery, but we will look down into the beginning of the brachial artery as well in this dissection. The axillary artery is that segment of the artery that is the continuation of the subclavian artery, and it begins at the lower border of the first rib, which is about here, even though we've actually removed the first rib at this point. And it runs from the lower border of the first rib all the way down to the point where it crosses the lower border of the teres major, which is about here, as you can see by the tendon of the latissimus dorsi muscle.
So it's already, if can you see as he's pinching it, it just snaps back, right? When he grabs it with his forceps, it totally just like returns into a tube. Or can does that come across on the video? Yes, no?
A little
Can you
bit.
go back?
Maybe a little bit. I don't know.
So here, let me look at it again.
The point where it crosses the lower border of the teres major, which is about here, as you can see by the tendon of the latissimus dorsi muscle underneath. So this would be the segment that includes the axillary artery. Now, it's nice to the tendon.
There, look at that.
And it runs from the lower border of the first rib all the way down to...
Like the vein does not do that at all. A vein is just totally super soft and flop. You might even rip the vein if you did that to it. This thing is this kind of rubber hose, and when you squish it back and forth, it actually just snaps back. Even more than that, it's actually kind of cool on this. What is this right here?
The aorta.
Yeah, so here you can get a sense. The aorta really is about the diameter of a garden hose. It's incredibly huge. And then, there we have like the brachiocephalic trunk coming off of it and all that. We'll come back to this. But here again, we've got the subclavian. This is the left subclavian into the left axillary. And this whole thing is very kind of thick and rubbery. Again, yeah, you have to do your best just to imagine it. Imagine it as like a rubber hose. Let me clear this.
To the point where it crosses the lower border of the teres major, which is about here, as you can see by the tendon of the latissimus dorsi muscle underneath. So this would be the segment that includes the axillary artery. Now, it's nice to divide the axillary up into 3 parts.
We don't care about that as much.
And 3 branches off the 3rd. I mean, is a very small branch, which is oftentimes torn. See those 3 branches here, a very large one.
So one thing I should mention, when you're studying, there are so many blood vessels. Your textbook and your lab book have so many blood vessels. You have a very limited number that you are responsible for. So make sure when you're studying, you use your, blood vessels to know list. Because if you just accidentally start trying to memorize all of them, you're going to get totally overwhelmed. So go back to your, your study guide and that page that says blood vessels to know, and make sure that you use that as a guide when you're like highlighting in your book or whatever, the ones that you actually need to memorize, because there are a buttload of vessels coming off of everywhere.
This sort of curly artery is the subscapular artery. The subscapular branches, dorsal. The other branch.
Yeah, I'm trying to get back to the brachial.
Brachial artery has a branch that is easy to find because it.
So I'm going to leave this one, but just to let you, if you look at this, you can just see it kind of holds, it's round, it stays looking like a big pipe. The next one I'm going to show you is actually pretty cool. It's showing the heart. So here in this one, they've taken the heart out, but it's kind of interesting to watch them take the heart out. Because we talked about the whole idea of the pericardial sac and all that. And it's going to be, I think it's worth looking at. So if we go back to this one here at 3, and it also is going to show the phrenic nerve, which I really think is cool. So this one, we're going to go to start at 3 minutes. She kind of goes in first and like cuts out the lungs and stuff. Takes off the sternum.
In the previous dissection intact, the internal thoracic or internal mammary arteries, which not only supply the chest wall, but also continue down onto the abdominal wall to provide collateral circulation there. Now with the bony part of the rib cage removed, we can see very nicely the 2 lungs on either side within the chest and the heart right along the midline. And we have preserved the internal thoracic artery and vein on either side in this situation. So during the next step of the dissection, what we're going to do is remove the lungs. And you should be careful to, again, preserve the internal thoracic arteries. And another structure that we hope that you can keep for the entire semester if possible, and that is a nerve that you'll find traveling inferiorly to reach the diaphragm, which it innervates, and this nerve called the phrenic nerve.
So this is actually pretty easy to see in the cadaver. It's easy to see in the pigs. It just looks like a little white thread going down along the side of the heart. So what plexus is giving rise to the phrenic nerve?
Cervical.
Yes, the cervical plexus. So this is coming all the way up from your cervical spinal nerves, going down along the side of the heart, down to the diaphragm.
Travels between the lung and the heart. Okay, you're going to find one on the right and one on the left. So that as you progress to removing the lung and cutting it out of the body, make sure you know where the phrenic nerve is at all times and keep it out of the way so that we can keep that intact.
So that is kind of cool to actually get to see the phrenic nerve there. And now I'm going to go to where she is going to actually show the heart in its pericardial sac.
Called the pericardial sac into this region and withdraw that fluid without the risk of puncturing the. Now that we remove the lungs, essentially everything that you see remaining here in the midline of the chest cavity is in a region called the mediastinum, and this would include the heart. Now the heart, like the lungs, is surrounded by a sac called the pericardial sac that will reduce the friction as the heart enlarges and contracts again. And I've got the scissors holding on to the outer layer of the parietal pericardium, and to expose the heart itself, you need to cut through the parietal pericardium, and we'll see the visceral pericardium right on the surface of the heart. As you do this, once again, be careful not to cut the phrenic nerve innervating the diaphragm, and try to keep those internal thoracic vessels intact as well. So I'm simply going to make a large X-shaped cut within the pericardial sac and flip back those flaps to expose the heart.
So, yeah, so there you can see it's really just sitting in this little bag there. And again, that pericardial sac is both the internal, the parietal layer of the serous membrane, as well as some reinforcing connective tissue to make that fibrous pericardium, which is why it's more of like a strong bag.
And you are now looking at the visceral pericardium surrounding the heart. You'll notice there is a fair amount of adipose tissue that's just deep to this layer, and we're going to have to dissect through that to expose the vasculature of the heart, the coronary vasculature. The next step is to remove the heart from the mediastinum by cutting through the great vessels, the major inflow and outflow vessels for the heart. Now I can start by pulling the heart superiorly and cutting through the inferior vena cava, which is bringing the blood back from the regions of the body inferior to the diaphragm.
Yeah.
Okay, and there's really no length to the inferior vena cava. It comes through the diaphragm and immediately enters the right atrium of the heart. Now on the superior end of the heart, I can put my finger through and cut through the aorta and the pulmonary trunk. And do make all these incisions within the pericardial cavity to make sure that you don't damage any structures that we want to have you look at later in the dissection. Again, I'll take a scissors and cut through the aorta and the pulmonary trunk. So those major outflow vessels have now been cut. The last large vessel that I need to cut through is the superior vena cava, returning blood from the upper limbs and the head back to the right atrium. Again, I will cut through that and the heart will be free. We've already cut through the pulmonary veins when we removed the lungs, so we just need to bluntly dissect those from their penetration through the pericardial cavity so that we can pull the heart out.
So again, just kind of reiterating, look at the size of the aorta. It is it's a garden hose.
We've put the heart and the sternum back in place on the chest so that you can get an appreciation for how much the heart is protected by the sternum and by the thoracic cage. Now you'll notice that the heart is positioned asymmetrically within the chest so that on the right side, it projects approximately an inch toward the right from the edge of the sternum. It's important to have an idea of where the heart is positioned so that you can predict whether or not the heart is going to be affected by a puncture wound or other type of wound to the anterior chest wall. If you did get some sort of an injury in this position, it would be the right atrium that you see here that would be most likely affected. On the left side, we see that the heart projects more like 3 inches from the left edge of the sternum, and it's the right ventricle and the left ventricle that potentially could be damaged or punctured by a wound to this region of the chest. Okay? We've got a nice landmark here to show us the boundary between the right ventricle and left ventricle. This is called the anterior interventricular artery, also known as the LAD clinically, and that is going to mark the boundary between the right ventricle. We can look at some of the features on the external.
So this is actually kind of nice. Here she's cut away a lot of the fat So you can actually see the coronary arteries. You know, for our class, you should know there's the right and left coronary arteries coming off of the base of the aorta. You're not going to be tested on the interventricular artery or the circumflex artery. I mean, they're kind of fun to learn about, but you won't be tested on them. This one is also at that LAD. This is the one that's commonly blocked in heart attacks. This one is often bypassed in cardiac bypass surgeries where they, again, maybe take some of your great saphenous vein and they will bypass the blocked part of this interventricular artery to keep adequate blood flow to the heart muscle. So she goes on. I think it's fun to see how, trying to get my little annotator again. You know, the veins tend to be kind of floppier and thinner. Arteries tend to be, this is, it's so big, it's bending a little bit, but it's got really, you can see how thick the walls of the aorta are here. So the other ones I'm going to show you. Is this interesting for you to kind of look at these pictures, these videos?
Yeah, I think it's super cool.
Okay, so I'm going to show you a couple of more little things that I think are interesting. We're going to go to 20, go to the femoral artery.
Surface of the heart, identifying some of the major vessels and the position.
Okay, back to here. We're going to go. This one I have 1330.
This is the section of the muscles of the hip, thigh, and related vessels.
Yeah.
And from the lateral surfaces of the vertebra, and continue down to form a common muscle belly, the iliopsoas muscle, which is the femoral artery nerve, and also by the obturator nerve. It also performs the functions of adduction as well as flexion. So it really can be put into either of these two groups. Now we're going to finish off the dissection by looking at the blood supply to the anterior thigh. And the main artery that courses into the thigh is the femoral artery, which is a continuation of the external iliac after it passes underneath the inguinal ligament, which would have been approximately here. As soon as the femoral artery passes across the inguinal ligament, it branches into a number of important branches. A lateral branch.
So you don't need to know any of these branches he's talking about, but I kind of want you to see, again, it looks like, I mean, I don't know how, it's always hard to know how much it looks to me different than you because I'm just used to seeing it in real life. So I just add in what it looks like. But it is this big rubbery hose. This
circumflexes the femur.
And we don't care about all those. Well, here you can see where it continues to the popliteal.
We'll follow down the adductor canal and pierce the adductor hiatus in the adductor magnus muscle to gain access to the back of the knee joint where it changes its name to the popliteal artery.
So there you can see the femoral artery kind of diving down behind to become the popliteal. And then we saw after the popliteal, it becomes the posterior tibial and has a little thing going forward with the anterior tibial and the thing off to the side for the fibular. But just to kind of give you a sense of how it looks, I'm trying to see if there's, can we see that?
The last branch.
Yeah, so one more, I'm going to show you one more just to show you how messy the stuff can get when you get into all the abdominal viscera.
pancreas. And of course, again, this is the root of the celiac trunk with the splenic artery and the common hepatic here. And they are emerging from the aorta just above the pancreas. The superior mesenteric artery emerges from the aorta just inferior to the pancreas, and that's shown here along with the superior mesenteric vein. Here's the superior mesenteric vein. It's just on the right side of the artery, which is located right here. Now, the distribution of the superior mesenteric artery is actually best visualized after you've dissected out the main stem of it as it comes off of the aorta. We want to take the large bowel, this would be the transverse colon here, and we're going to flip it up. And you can see, we can still see the stem of the superior mesenteric artery, but now each of the branches of the superior mesenteric artery is really quite evident. Now, once again, this is the cecum that...
Right, so here you can see that superior mesenteric artery actually starts serving all sorts of your intestines here. This video is harder to see. Everything looks kind of just all bloody.
Large branch that heads right down towards the cecum. Actually,
let's see.
Down, and here are all of these anastomosing branches. Ever so much so that we can see the pancreas, which is right.
Well, I think that's it. Let me close this. So there's probably more stuff. This is stuff that I found this morning. Again, just trying to give you a sense of what these things look like in real life versus like in these pictures. Let me stop the sharing. So what time is it? It's 4, a little after 4. I was also looking to make these Pearson homeworks for you this morning and realized it's going to take me more time than I thought I could. I thought I could just like whip them out. And instead I realized every question I have to kind of go through, like, what is this question, and go through it. And does it seem like a good question? And so it's going to, my plan is before today is done, I am going to add in some Pearson assignments. Let me share this again. So I will be creating I have not done them yet. They will be really useful, I think, because images that I'm going to be drawing from for your exam are coming out of this system. So for better or for worse, doing these Pearson things will be a useful way of studying for the exam, because it's part of what I have to do testing now that we are in this virtual world. So I will definitely send you an announcement, and I will put little information in modules about what is available and when it's due and all that. So it will come into being, and I will make sure you know that it is now in being. What else? I'm trying to think if there's. I think that's
I have a question about will we have access to see what we got wrong on the quiz?
Oh, duh. That's what we're going to do. But so you can't see can you not see what you got wrong on the quiz right now?
No, all the answers are hidden currently.
Okay, so stay with me here then. So let's go over the quiz. That's what we'll do for the rest of today. We'll go over the quiz, and I am also going to figure out how I let you, like for the main exams, I won't do it. But for the quizzes, since I usually let you just keep the quizzes, I most definitely want to figure out how to do this. So I'm going into my modules, going into anatomy quiz. I'm going to edit it. Okay, let students see their responses. Let students see, show correct answers right now. So you're in front of if you're in front of your computer, can you check to see if that did it?
Yeah, that worked.
Okay, so let's go over it though, since I have not typed this all into my my other program that lets me do like how many people got A's, B's, C's, and whatever. And Canvas does not do a nice statistics. I mean, I could show you the basic statistics. Quiz summary, come on. You know, we got, I don't know if, can you see this? Is this on the screen for you guys? This little graph here? You can see.
Yeah,
like,
we can see it.
Yeah, so this was like kind of 90 to 100. Most people were in this zone. Some people here, we had, well, we won't talk about that one. So let us go and go back into the quiz and just go one by one. We're gonna, I guess we can kind of preview it. So the first question was polio. And it's saying, so this used to be, you know, now we're worried about the coronavirus. Like I had teachers when I was in college who talked about how every year when they were kids, after summer break, they would see who got hit, who's going to be paralyzed. It was not uncommon for your kind of schoolyard play friend to end up getting paralyzed at some point because they caught polio. Worst case, it could actually paralyze, you know, make it so you couldn't breathe without being in an iron lung, like a thing, basically a ventilator. So it causes paralysis. And so which part of the spinal cord is going to have to do with motor?
The ventral horns.
The ventral horns. Central canal obviously is just a hole with CSF. Dorsal root ganglia is not in the spinal cord at all, and plus it's sensory. Lateral horns were that sympathetic, and dorsal horns are sensory, so it's ventral horns. Moving right along. And I should, so some of these things are randomized. There's different versions, so you might have a slightly different version. So if your question is not exactly one of these, it's a related one. Primary somatosensory cortex. Where do we find the somatosensory cortex?
Right behind
Placenta,
central.
gyrus.
That's going to be post-central. Remember I was saying it's like I think about it in terms of alphabetical order. Motor comes before, M becomes before S. So S is later, S is the post one. You know, precentral would be my primary motor cortex. This one I threw in here just because it rhymes. I didn't even, as I was kind of coming up with it, it's like, oh, coronavirus gyrus. It's kind of rhymes. The falx cerebri is composed of
Dura matter.
it's the dura mater. It's one of the dural septa. It's the dura mater that separates the right and left cerebral hemispheres. So then we can continue on. And lateral ventricle. So where do we find the lateral ventricle?
Cerebral hemispheres.
That's the one that's in the cerebral hemispheres that gets all, extended out into that big weird C-shaped looking thing. And then third ventricle.
In between the thalami.
Yep. The right and left thalamus is the two walls of the third ventricle. The glial cell that makes myelin sheaths.
Oligodendrocytes.
Oligodendrocytes. Continuing on. Function of a choroid plexus.
Produce CSF.
It's making the CSF. How about I think another question was like arachnoid villi. What's the function of the arachnoid villi?
Drain the CSF.
Maybe to drain the CSF. Yeah, I was pretty proud of myself for finding this picture here. So double vision can result from improper functioning of the extrinsic muscles that move your eyeball. So you just have to remember which are the ones that do the muscles on your eyeball.
3, 4, and 6.
Yeah, 3, 4, and 6. Oculomotor, trochlear, and abducens. A lot of people put, the one with 2 and 10. That is not going to work. 2 is sensory. 2 is eye, but it's the sensory. The optic nerve is taking information from the eye, taking it to the brain. It's not actually operating muscles on the eye. So it was 3, 4, and 6. White matter in the brain is composed primarily of
Myelinated axon.
totally myelinated axons. I need this is a typo if you're watching petting. If you were petting a soft fluffy cat, and I did say, where would the tactile touch information be processed if you were having the tactile information from the cat?
Parietal.
Parietal. Parietal, that was where you have that somatosensory cortex. I had different versions. I said, what if you were looking at a cat? What lobe would it be processed, the visual information, if you're looking at the cat?
Occipital.
That'd be occipital lobe. And then there was another question that said, what would happen if you were listening to the cat meow?
Temporal.
That would be temporal. So those are all different possibilities there. Brain injury that damaged the prefrontal area. What would that cause?
Loss of impulse control.
Impulse. Prefrontal was executive, impulse control, all that. I think another version of this question said, what happened if you damaged Broca's area?
Speech production.
That'd be speech production. And if it was Wernicke's area?
Comprehension.
Yes, inability to understand language. So. The ganglia of the parasympathetic division. Where do we, which answer is right?
Close to the target.
Yeah, these are the ones that ganglia are close to the target. We talked about the intramural and terminal ganglia. So definitely fight or flight is parasympathetic. The parasympathetic ganglia are close to the spinal column. The sympathetic chain is the one that goes up and down. It's not going to be ganglia if it's within the spinal cord. That's ganglia by definition is not in the CNS. So it's going to be located near or even within the wall. This one, some people got confused. Ventral roots carry what kind?
Motor.
Motor. And dorsal?
Sensory.
And then what about the spinal nerves themselves?
Both.
Both, right? Because both the ventral and dorsal roots merge together to become the spinal nerve. And again, I use the same mnemonic I use for the pre and post central gyrus. You know, M comes before S, so M is the one in front, ventral towards the belly, dorsal is towards the back. So S is further back in the alphabet, so it's the dorsal roots which are in the back, and M is closer to the front of the alphabet, which is ventral towards the belly. So I don't know, you can use whatever tricks you find useful. This question maybe I didn't ask. Some people got what I was asking and some people really did not. What I was really hoping is, prosencephalon. You know, what structures in the adult brain? It's like the cerebrum, you know, the diencephalon, the hypothalamus, and the thalamus, and you know, stuff like that. You know, mesencephalon, that one we just kind of call it the midbrain in general. Although we could also use, you could say like substantia nigra and corpus callosum. Rhombencephalon, you know, I really wanted there to be a cerebellum and a pons and a medulla oblongata. Some people just used, you know, kind of, you know, if you said midbrain is kind of right, but if you just said prosencephalon is the forebrain isn't like a structure of the adult brain. So I only gave full credit on this one if you did something more like what I wrote here. I did different amounts of partial based upon how much you got of this, or kind of, you know, I gave people some credit because I figured maybe I could have wrote it a little differently, but you know, saying prosencephalon becomes the telencephalon and the diencephalon. It's still not quite the structures. You know, diencephalon is kind of, we talk about the diencephalon as a area, but you know, so I don't know, it was tricky trying to figure out how to do this one. All right, name the gyrus labeled B.
Precentral gyrus.
That's the precentral gyrus. As you can see it kind of in the front. And, you know, I felt okay on this picture because the central sulcus is so well defined, and then it's going to be, you know, or you could say motor functions or motor whatever. You know, this one, what is the structure labeled B?
Cingulate gyrus. No.
That was actually the corpus callosum.
Callosum.
Yeah, the cingulate gyrus would be right above it.
Right above it.
You're totally in the right spot, but the thing that's right above the septum pellucidum here is this is the corpus callosum. And is it white or gray matter?
White.
White matter.
It's white because it's a bunch of axons. It's a major tract, a commissure. And then here we got the, you know, it's pretty obvious, the biceps brachii muscle with the two heads. What nerve innervates the flexors of your upper arm?
Musculocutaneous.
Musculocutaneous.
So this is where, I was kind of saying you need to be able to, don't forget your muscles, even as we're going on to other things. You still got to know what muscles are. What's this big honking nerve? That's the sciatic. Coming out of the, we can see we're on the back side. It's coming out from the sacral region. If we had him flipped around and we had it coming from the lumbar region, it might have been the femoral, but because we're on the back and coming from the sacral region, we know it's the sciatic. Here on the model, again, we look for the M, and we have the middle. What's the thing coming off of the middle part of the M?
Median.
Median nerve. What muscles would be innervated by the median nerve?
The forearm flexors.
Yeah, the forearm flexors. But basically, everything except for flexor carpi ulnaris is going to get that one. Here I have a nice little picture of a spinal cord. What area is indicated by the red arrows?
Dorsal roots.
Not dorsal roots.
Dorsal horn.
Dorsal horn.
So the roots are the nerves that are actually coming out already. So if I was looking at the axons that are, this is the roots. And they actually are going to go coming into here where we have unipolar neurons. What is this structure over here?
Dorsal root ganglion.
That's the dorsal root ganglion there. So let me clear up my drawings, get rid of this. And what kind of information is processed in that dorsal horn?
Sensory.
It's going to be sensory. So sensory information is coming in there. Now we continue on. So this question, I basically says, name at least 3 cell types present. So the main thing you just need to know is, oh, I'm looking at the spinal cord. So I'm looking at the CNS. So what are different cell types you might find in the CNS?
Astrocytes.
Might be astrocytes. What else?
Neurons.
Neurons.
Microglia.
Microglia.
Oligodendrocytes.
Oligodendrocytes.
Ependymal.
Video.
Huh? The ependymal cells are going to be in the little central canal. So any 3 of these, instead people started putting like satellite cells and stuff, and those would only be in the PNS, not in a picture of the CNS. What is that big sheet that the red arrows are touching?
Falx cerebri.
Yeah, that's the falx cerebri. Hollow space indicated by that arrow.
Ventricle.
That's the 4th ventricle. This is where you got to be careful. Make sure you read the question. Some people wrote thalamus or something. They didn't read closely. Name the hollow space indicated by the region. Which one is this going to be?
Third ventricle.
Third ventricle.
Going to be the third ventricle. And this is nice. It shows a nice corpus callosum, septum pellucidum. This one says name the structure. So this is then going to be the thalamus. And the function, there's lots of functions, you know, sensory relay or something like that. Here I was like trying, you know, name the overall structure. I kind of meant this whole thing, but in reality, I probably should have put some dotted lines around it or something, because some people said arbor vitae, which is kind of the specific axons in there. What is this overall thing called?
Cerebellum.
Cerebellum. And then function, what is the function for the cerebellum?
Smooth coordinated movements.
Yeah, smooth movements. Yeah, there's lots of things. Smooth coordinated movement. Lots of things you could say there. Just about kind of coordinating motor. Then the final one. So this is a nerve. You can see all the little axon cross sections, all the little donuts here. What is the fibrous connective layer that is the outermost coating of a nerve?
Epineurum.
It's the epineurium. And then, is it CNS or PNS?
PNS.
So this one, a lot of people got this wrong. And you kind of had the answer in the question. It tells you that it's a nerve. And by definition, a nerve is a collection of axons in the PNS. So, you know, so you want to make sure you slow down. Like, so if you know it's a nerve, then you know it's got to be the PNS. I'm submitting the quiz. And yeah, so obviously, I think it's kind of silly they even give you these partial scores, because it just makes people feel nervous. Whatever that score is there, it's the one that it automatically was able to grade. Although sometimes people get things right, they just didn't type it in exactly the way I typed in the answer key. So I go back and you did get it right, even though it makes it look like you got it wrong. The short answers aren't graded even to start with. So you can't really trust that number meaning anything. So I really encourage you not to let that thing mess with your head. I kind of wish it didn't even print out like that, because it's just gonna make people feel like they failed or something. So are there any questions about all of that?
So in regard to the setup, when you had us like not be able to go back and look at any questions, are you going to keep that for the exams?
For the exam, you can't go back. Yeah, that's again from the stuff I've been reading about trying to do online testing. That's the setting they recommend. So I am going to do that.
Would it be possible to give more time? And that was a part that was really challenging, especially knowing that you couldn't go back.
Yeah, no, for sure. So, I mean, that was good information for me to find that out, and I will do my best to work with that and try to make sure you don't feel stressed around time.
Just something to consider in other online courses that I've taken. There's a feature to flag a question, and then you're able to go back and see it afterwards if you're kind of stumped on it. I don't know if that's something you would consider incorporating.
I don't know if that exists. I haven't seen or heard about that in Canvas. I can look into that. Let me take a note for myself to look into that.
It was on Canvas when I did that, so I believe it is a feature of Canvas.
So, so then maybe it has to do with, there's that setting that I have. Yeah, so you show one question. Yeah, so if I lock the questions after answering, then it can't let you go back. Yeah, so no, so I probably can't do that. If I lock the questions, then you can't, even if you flag it, it's locked, so you can't go back. So probably if I unlocked it, then you could do that, but then people could go back and forth, and yeah, I think I'm going to keep it the way it is.
I mean, just in my opinion, I would prefer to be able to go back because I know like sometimes after I click the button and I was on another question, I remembered the answer, or like I knew that I answered one before it wrong.
So I do know that happens, and I think we just have to like work with this, and it'll go into the like seeing how people do on these tests in general and how things get curved and all that as well. But like I said, I didn't kind of make that up. I've been reading all these best practices for online testing, and in general, they recommend doing it this way. It gets tricky because you might be completely doing the right thing, but then other people are doing things with their friends where they're trying to go back and forth. Or I saw this question, now I'm going to go, this is what, so it's unfortunate, but there is basic human nature that we have to try to work with as well, and trying to do this online testing to keep it at least kind of sort of secure. So that's where that comes from.
Thank you for putting all this together.
Sure. And
Yeah, bad.
oh,
Sorry.
no problem. So are there any, I just, while we've been having our class, I've been rendering Tuesday's lecture and I'll have that uploaded on YouTube, and I will also have today's lecture uploaded on YouTube before the day is done. And I'm starting to take stuff, taking it off of the Confer Zoom site and just leaving it up on YouTube. So I'm not using up the memory on the school server. So you'll definitely wanna, if you wanna find access to these different ones, you'll find them on YouTube, on the YouTube channel. So, and again, I will be spending time this afternoon and evening, because it's almost evening, coming up with these Pearson assignments. So I'll give you these Pearson assignments, so that will help you review the blood vessels, review the heart and stuff like that. And I think that will be helpful, something you probably, you and me are probably all, you read these things about people, so I'm so bored, what do I do with this shelter in place, it's killing me. And I'm like, oh my God. It's like, that sounds so nice. But at least you guys have something to keep you busy while you're sheltering in place. I guess the silver lining. So I will see you all on Tuesday.
Okay. Thank you, Dr. Eagert.
Thank you.
Thank you.
Have a good weekend.
Thank you.
Bye-bye then.
Dr. Eagert, this is Casey. I had a quick question.
Sure.
I think on one of the quiz questions, it didn't kind of check me on one of them. I think I got it right.
Here, let me take a look right now. Hold on one sec. Again, I'm also getting used to grading in this. Yeah, I cannot say I love Canvas. So I'm going to go, give me a few moments here. I am getting quiz number 2. There it is, and Casey Warner.
It was on question 23. I put in the Roman numeral third ventricle and it marked me wrong on that.
Okay, so let me take a look here. So we're gonna go into SpeedGrader. I'm gonna share the screen so you can see exactly what I'm seeing. So hold on one second. Actually, can you see the screen right now? Can you see
Your
quiz for Casey Warner?
Yes.
Okay. So right. So if you take a look what happened here, it says, you know, wrong.
Question 23.
You see this? I went through and I saw that that was just fine. So I just upgraded this to 1 out of 1 points.
Yeah, I think you caught most of them. It was just for question 23. I don't think you
Let's
caught
go down to 23. Hold on one sec. Oh, you're right. You're totally right. All right. So 0.5. And we go down here to update scores. And you are now at 23.16 points out of 24, which is pretty awesome.
Thank you so much. I appreciate that.
All right. Yeah, no, thanks for catching that. Cool.
All right. You have a good weekend. Thank you.
Bye-bye.